Good afternoon, madam.
Good afternoon, Maheshwari.
Yeah, good afternoon, madam. Good afternoon.
Also, ma'am also has joined this
Yeah,
session.
Kausi madam has joined, ma'am.
Yeah.
Yeah. Good afternoon, Kausi madam. Good afternoon, students. Please inform to your friends. Let them join as early as possible. YouTube live and Facebook live is also going on. Please check it. You can join at there also. Lakshmi madam, within 2 minutes we'll start the section.
Sure. Yeah.
Did Kausi madam is ready?
Yeah, she is ready. I think, yes, she went out. She'll join.
Yeah, okay. Yeah,
Mike,
okay, ma'am.
hello.
Yeah, ma'am. Good
Yeah, good
afternoon.
afternoon, madam.
Yeah, I'm here. Good afternoon.
Yeah, okay, madam. Within 2 minutes, we'll start the section,
Yeah.
madam.
I'm just going to get my official laptop so I can go through the presentation while you can start. Right?
Yeah, okay, madam.
Are our listeners all joined?
Just they are joining, madam. Students are
Okay.
joining.
No problem. No problem. Okay.
Ma'am, you can also enable your video for. So, Maheshwari?
Ma'am.
Yeah.
Ma'am, can we start? Good
Yes,
afternoon.
Maheshwari, we can start the session.
Yeah, okay, ma'am. Good afternoon, students. Today our speaker is. Can you audible, student? Good afternoon, students. Today our speaker is Dr. Kausimani. She completed his PhD and he is working. She is working from 2018 February in the Dr.Reddy's lab. Madam, heartily welcome to the section. You can start the section and you introduce yourself.
So, actually, Maheshwari, if you can project the slide.
Yeah, I can,
And
madam.
today the first 1 hour would be taken up by me,
Yeah, okay, ma'am.
and the next 1 hour would be taken up by Kaustubh ma'am.
Yeah, okay, ma'am. Please send me the PPT, ma'am.
I did share you.
Yeah, okay, ma'am. I got it.
Yeah.
Give me 2 seconds, ma'am.
Sure, sure. So, good afternoon, everyone. I hope everybody are waiting for the session today. And this session that we will take up today is, very much connecting to your, lab activities that you're doing in your BSc, MSc, BPharm, and MPharm. So I am sure you guys would be more intact with us throughout the session. And any questions, as usual, you can drop it in the chat boxes. Yeah.
Madam, it's downloading. Please.
Okay. Yeah. So, on introducing myself, I am Bharatalakshmi. I am a faculty at Dr. Reddy's, and I do trainings to the quality control employees of Dr. Reddy's. And over the last 3-4 years, whenever we used to train new joinees, we always used to feel there is a gap in the understanding level of the students. So this kind of sessions will make the students know the topics before they are landing into the premises of any organization. And we even Kaustu ma'am is part of technical training. Yeah. And madam is also taking up sessions for the quality control employees. And She carries a lot of knowledge with her. She's a PhD, and any questions, guys, again, you can put it in the chat boxes. So, I hope everybody can
He
hear
asked,
me. And
Madam, PPT is asking password.
okay, you can open as read only, right?
Yeah, okay, ma'am.
Is this the one? I'm not sure. Okay, I think this was not the one. Okay. What I will do is I will try to project the slide from my side. Will that be okay?
Yeah, okay, ma'am.
I think until you, yeah, that helps. So whenever there is a question, Maheshwari, just let me know.
Yeah, okay, madam. I will read out for you.
That helps. Thank you, guys. And shall we start the session? If you want me to start the session, you can say yes. Let me see how many
Students, please respond.
chats. So are they ready, Maheshwari?
Yeah, yes, ma'am. They put
Yeah.
yes also.
Okay, great. So I hope you guys can see the screen also.
Yes, ma'am. Yes,
You
madam.
can, yeah, right. So moving ahead with today's presentation. So it is on pharmaceutical analysis. So let's understand what in the next 2 hours, what we can learn in this 2 hours time. So here we bring in to you that there is something on QC essential, and there will be a second section by Kaustubh ma'am on analytical techniques. And let's in QC and essentials we'll be talking about what are the various functions of quality control, what are the various regulatory agencies, what are the various guidelines about pharmacopoeias, and you must have also heard about something on GLP. So we will talk, we will discuss on that also. We will specially focus on, there are computerized system requirements. Now, and then alcohol, reagents, chemicals, solvents, glassware which is used in a laboratory environment. What are the standards? Again, laboratory standards which are used here. Again, covering instrument equipment. So this is a brief. In the next 2 hours, we will be talking about these topics. And any questions, you can drop it in the chat boxes. So to start off, I just have one question. What is quality to you guys? What does quality stand to you? And it may be anything, whenever we are buying any, new dress or new shoes or new bike or new car, we always see whether it is quality or, whether it will last for 2 years, So, if at all you can just message in the chat. What do you any word which is a, which says this is quality to you? Any responses, Maheshwari?
No, ma'am.
Okay.
Just I dropped the question.
Okay.
Stands present in the limits, in with
Oh
limits.
wow, wow! You are very well, going in depth of pharma, and yeah, you are right. Absolutely, there are something called as limits, and there are specifications. Of course, any product that, especially the tablet or syrup, which we get in the market, will have a specification, and if the specification is meeting. That is again, very well, whoever it is, typed in the chat box. Thanks for the response. Limit. Yeah. So if a product is meeting the limits, and again, there are a lot of tests here that we perform in the quality control labs. It is not just one test that we do and we say it is good and we release it to the market. It's not like that. There are a lot of tests. And again, I'll just try to give you an example. Suppose if we want to, organize a birthday party for our friend, and or I am giving a party to you, to all of you, then I look up a restaurant which is, which not only delivers good quality food, that means the taste, the number of items that are coming in the menu, and then the space in the restaurant, all that are looked upon by me when, and I see, this is where I want to, bring all my friends for the birthday party or event. So there are various aspects that I am looking. Similar way, if I am sending my, releasing, the word here in QC is releasing the product to the market. I also look upon lot of tests. Lot of tests are done, and then only based on the product meeting the specification, we release the product. Now let's go in depth. So there are, again, it is not always that I approve the product. There are days, if the product is not meeting the specification, definitely, as a quality control personnel, I would reject the material and it will never be sent into the market. So both, and this is very important job to anyone to decide, the fate of the product. Now, a lot of money is invested into the manufacturing process, whether it is a chemical operations like API product or a finished product, or an injectable. Every product has to undergo a test in the quality control, and then it will be released to the market. So there are, there are a lot of regulations around, and a lot of guidelines around this. So there are definitions like, checking or testing the specifications are met. So this is clearly given in ICH Q7. ICH stands for any responses. What does ICH stand here? So you can drop the message. We will come back on that. There is a definition on quality control. Quality control is concerned with sampling, specifications, and testing, as well as the organization, documentation, and release procedures, which ensure that the necessary and relevant tests. Okay, every word has its own, importance, and that's where I'm reading the definition. Are carried out, and that materials are not released for use, nor products released for sale or supply until their quality has been judged satisfactory. So this definition is taken from Udrelex. Okay. Now again, you guys know what is CFR. So again, very important, we have something called as FDA. If you remember the first session where Mr. Beda was talking about FDA, Food and Drugs Administration, and again, the agency has released regulations which are called as Code of Federal Regulations. It is abbreviated as CFR for us, and this CFR has lot of regulations, and 21 is a chapter where we refer to it, and 21 CFR 210 and 211. 210 and 211 are more focused on the drug manufacturing products. And there also we have a very good definition under the subpart B about responsibilities of quality control. More or less, it is about approving or rejecting based on the tests. And in the same, 21 CFR, if you go to the subpart I, there is something beautifully talked about laboratory controls. So you guys can go through the contents which are there online. So let's come back. What are the functions of quality control? So, broadly, the activities which are done in a QC, quality control lab, they are sampling, sample handling, testing, test reports. It is not just always testing, but there are other activities. If you see here, there are something called on instrument calibrations. So instrument calibrations, validation of samples, and method validation, and also microbiology is part of quality control department. So again, each function, I'm sure the name itself is talking about what could be the role. If we are saying sampling, definitely it is talking about sampling of the product, whether it is an API or a finished product. When the process is manufacturing process is going on, we pull up the samples during the manufacturing operations, and those samples are given to QC for testing. And not only the products, but there are if you see, there is sampling of air, sampling of water. So again, in the manufacturing, we use purified water, though that purified water also has to undergo a process of testing in the microbiology lab. So there are a lot of tests for purified water also, right from pH to TOC. Whether the purified water which is used in the which is used in manufacturing is without any kind of microorganisms like Staphylococcus aureus, etc. So that is also one of the aspect of testing. So, sample handling, retention, testing. Testing is a major chunk of a quality control department. Right, they analyze, raw materials, packaging materials, in process products. So in between, not even the finished product, but there will be kind of mid-time products are also taken for the testing. So again, reaction mixtures, finished products, stability samples. Again, after the marketing of the product, the samples are also loaded into a chamber for stability testing. So to determine the expiry date, shelf life of the product. So every product has 2 years of stability, or there will be some products which have 6 months of stability. Now, how does that timeline has tagged to the product? So that is again a very interesting area, which is stability testing of drug products and drug substances. So again, the testing part of those samples which are charged into the stability chamber is done by quality control. Then test reports, no doubt, test reports are important, and they typically, the hard copy, writing with pen is done on what is called as an ROA, record of analysis. But now most of the documentation is carried out electronically, so that any kind of lacuna in the document, like our handwriting is also one of the challenge where people cannot understand. So that's where electronic documentation will help us in all these aspects. And nowadays most of the labs are using this electronic documentation. We will come back on that. Then record maintenance, then instrument calibrations. Now we will have some definitions on calibrations and quality. Whenever we are testing a sample, it is not just the sample, but we test the sample with various instruments. I am sure you must have heard a lot of instruments say, your intermediate as vernier calipers, where you guys were testing, the length, width, etc. So those are the simple instruments which are used in the lab. Apart from that, you must have also heard about HPLC, GC, which are, high-end instruments. Those are also there in the lab. But whenever we are using them, they should be in a working condition. So that's where we have a, regular maintenance kind of thing for the instruments. So we will talk on that in detail. Then we have something on validation samples testing. It is again just like the regular sample testing. We will do analysis for the validation samples. And this line item, which is method validation. So I have a sample, I have an instrument. How do I test? So there should be a written procedure of testing my product. So that is called as a method. And this method is like, for us, if method is not there, how I have to do the testing? In your labs also, you guys definitely write about the objective for the test. What are the various requirements of the tests, like and then reagents, chemicals which are used during the testing. And you guys also document something called as procedure in your laboratory notebooks, right? Yeah. So that is where method comes, and the methods which are adopted for the releasing of a medicine, definitely that method should be validated, so that the method can be repeatable, reproducible, whether I am doing the testing or you like Gaurav or any other guys doing the testing. The final results should be repeatable, reproducible. And I repeat, the R&R, the repeatability and reproducibility is very, very important in our quality control. And so that's on the various functions of quality control. So any questions to take here?
No, madam.
Okay. So now, did somebody mention what is ICH? Okay. Now, you guys are,
ICHS, madam.
yeah.
International Conference on Horomethylation.
Okay. It's actually International Council on Harmonization. Okay, it's actually, guys, I tell you, you guys should go through the ICH website, and you guys should have a habit of, going through the contents which are there in ICH. Uh, why we should know? Because these guidelines help us in understanding how the manufacturing operations function. So and these ICH quality guidelines are we listed here, but there are 14 if you go to the website. The first guideline is on stability. So Q1 is on stability. Q2 on analytical validation. Guys, you have to remember we will have short quizzes in between, and I expect you to tell me what the answers would be. Okay, right. So Q1 is on stability, Q2 on analytical validation, Q3 is on impurity testing. Okay, so Q1, Q2, Q3. Q4 talks about pharmacopoeias. Q5 talks about biotechnological products. Q6 is talking about specifications. Q7 is talking about good manufacturing practice for API. And Q8 on pharmaceutical development. Q9 quality risk management. Q10 on pharmaceutical quality system. Okay. So at least, as an, if at all you are interested in quality control and you are looking forward for your career in QC, I recommend, you should at least do this first three of the guidelines. You should go to the website, download the PDF, and you should read once before, before your interviews in any of the pharma company. So these are various guidelines, and here is one question. I was talking about CFR. I said it is a regulation and we should follow it. I am also telling, there are something on ICH guidelines. We also should follow it. Now, what is the difference basically? What is the difference between 21 CFR and ICH guideline? Why should industry follow the guideline? So the answer to it is CFR is a regulation which is like thou shall. It recommends, it always says, thou shall, and it will ask us to follow those regulations. And guidelines, it's our acceptance. We can adopt those guidelines in our company or no, but regulations we have to follow it. Again, guideline, it's a very good procedures written very well, very refined, and lot of queries will be resolved for us if at all we are going through these guidelines. So guideline helps us, to uh, put our, steps forward and meet the regulatory expectations. So moving forward again, it's not just a 21 CFR, which is, to 10 to 11, which is from US FDA. It's not just one regulatory agency that we see in around the world. There are other regulatory agencies also, like MHRA. It stands for MHRA, stands for the Medicines and Healthcare Products Regulatory Agency, United Kingdom, and their guideline is Orange Guide. Uh, MCC, Medicines Control Council, South Africa, they have MCC as a guideline. ANVISA, ANVISA, it's for the Brazil market, so they have ANVISA guidelines. So the each and every, if you see, there are various countries with various regulatory agencies, etc. So TGA, Therapeutic Good Administration, TGA, Australia. The country is Australia.
Excuse me, ma'am.
Yeah,
From your background, some disturbance is coming.
yeah, it's the flight actually. It's the sound of the flight.
Okay.
Yeah,
Okay, ma'am.
yeah. So it's the next is CDSCO, Central Drug Standards Control Organization. This is for Indian market. So we have Schedule M, which is, if at all I set up a factory in India, I am supposed to follow this, guideline which is on Schedule M. So federal services. So again, you see a lot of European Medicines Agency, EMA. I'm talking, it's not kind of memory, but, when you actually happen to go to a company, then definitely you guys will know why so many are there. And if at all I am shipping my product, and then if I am marketing my product to US, I am also supposed to meet their, the country specific guidelines. So that's where we have to see. If I am shipping my product to Japan, I am supposed to follow, JPAL, which is Japanese Pharmaceutical Affairs. And worldwide, we have a common association, and also, ICH is one where, complete world accepts it. And similar way, there is something on PICS, Pharmaceutical Inspection Cooperation Scheme. So this is also one guideline. And actually, you must be wondering why, Lakshmi is talking about all these various regulations, regulatory agencies, etc. because my product will be shipped to that particular country, and I am supposed to ensure that my drug substance or drug product which is released to that particular country should meet what they want, what they are looking for. So that's where I should ensure that I'm meeting that. Then only they will accept my product, etc. So again, at Dr. Reddy, since we have various markets, even China, etc. We also have a QMS system. Our specifications, our STPs are very broad, and they are target countries. So based on the target countries, we have a lot of specification, lot of STPs, etc. So the same product, say I have a metoprolol product and I am shipping it to UK and US. So both of them, will have two different testing procedures. That is the testing methods, and they will have, different specifications also. So it is again, if at all both of them are same, then it's our luck. But usually there will be minor changes for country to country, but 90% would be a common practice. Okay, so I have some questions here for the youngsters to answer. Anybody? Which is not the function of QC? Anybody who can answer? Option A, it says sampling of raw material, PM, FP, raw material, packaging material, finished product. Option B, testing. Option C, generation of test report. Option D, retention of samples. Option E, review of BPR. BPR is a batch production record. What could be the answer? Which is not the function of QC? Any responses? If you are not going to answer, then our training will be delayed.
E, madam.
E,
Review
yeah,
of BPR.
correct. Absolutely. Review of BPR. Correct. Again, next question. Guys, you ready? I'm putting up the next question. I'm asking to match. So, GMP for API. Again, I was talking about ICH guidelines, Q1, Q2, Q3, like that. So here are some, on the right side, B column, you have Q1, Q2, Q3, Q6, Q7. If you can match them with, GMP, impurities, stability, analytical validation specifications. At least, one or two matches, if you can drop it in the chat.
3 Q1, madam.
Okay. 3 is, oh yeah, good. Great. Yes, you're right. Stability is for Q1, absolutely. Any other answers?
4 for Q2.
4 were Q2. Yay, good. Who was that? Good.
Shivareddy.
Reddy. Good job. Yeah. So I give the answers to you guys. So impurities is Q3, stability is Q1, analytical validation Q2, and specifications. Now I'm sure you will go to the ICH website to find what exactly is written in those guidelines. So again, one more match the following for you guys to play around. So country specific guidelines, we have European Union, United Kingdom, South Africa, Australia, USA, India on the left side, and we have right side as TGA, USA.
One for EMA.
Very good. Yeah.
6 for CDSCO.
Yes, good job. So I release the answers to you guys.
Fifth one is USFAD.
Okay, good. United States Foods and Drugs Administration. Yes. And India CDSCO. Great. So you guys are absolutely hearing me. So Lakshmi, for you, we'll move forward. Next content. Yeah. So Pharmacopoeia. Now, Not exactly. I am again pretty sure you all guys will have in your colleges, huge libraries where at some day your organic professor, or they must have asked you, go to the library, go to this particular pharmacopoeia, check for this acetyl salicylic acid, draw the structure and draw molecular weight of it, kind of questions. So with that, acknowledgement to the colleges, I move forward. So, what is a pharmacopoeia? It is a book or encyclopedia of drugs where formula, method of testing, specifications, etc. are published. And again, pharmacopoeias are changing based on the country. US has a USP, British UK has a British pharmacopoeia. So each and every country has written. Of course, India again has Indian pharmacopoeia. So in any pharmacopoeia, there would be the testing methods would be given for us, and it makes our life easy. Basically, every time we cannot invent a new method, we try to adopt the methods which are already there, previously executed and validated by so many organizations. It is already published, so we try to adopt that particular method. Not only that, we use a lot of instruments, right? So there are procedures which mentioned in the pharmacopoeias about the calibrations. What exactly is the calibration that should be done on my instrument? I have a lot of instruments like pH meter, IR, UV, spectrophotometers. I'm sure you must have seen in your analytical labs in your colleges. So those are also very well mentioned in the pharmacopoeias, and as industries we accept them, we ensure that our instruments follow or meet those calibration requirements which are written in those pharmacopoeias. So with this brief, we have countries. India has Indian Pharmacopoeia, USA has United States Pharmacopoeia, UK for British Pharmacopoeia, EU for has European Pharmacopoeia, Japan also has its own pharmacopoeia, China, and international pharmacopoeia also. So pharmacopoeial and also not only these, each there will be a renewal time. There will be and there will be supplements published by the pharmacopoeias too. So any questions? I hope no questions. Move forward. No. So
No questions, ma'am.
yeah. So right now I am in a quality control lab, and I just wrote, what are the various kind of practices that are done in a quality control. So on the left side, if you see, there is lab management. How the people should entry, how they have to enter, exit, their safety. Definitely, a lot of chemicals are there in the lab. So how should I, ensure that everybody is safe in that particular lab, and lab upkeep, lab behavior, etc. This is especially, like a lab management. I'm sure, you also have labs in your colleges, and you have lab technicians, etc. And you wear aprons, gloves, masks, etc. during your analysis, or testings, or during organic synthesis, etc. So those kind of practices only are normally seen in industry, but the rigor which with which we follow is on high side. So like you may wear a gloves or not wear a gloves in your lab, but when it is industry, it is like I have to wear it. So it's that. Now resource management. again, the employees are very huge number in the labs. So we have to manage our resources because a lot of testing should be done, etc. So materials, chemicals, reagents, solvents, etc. And instruments. These are all various resources that have to ensure that they are properly managed. If at all, I have a sample, I don't have people. Can the sample be released? No, definitely no. At the same time, I have very well trained analyst with me. I have instrument. I have sample. I don't have a reagent to test. Will it suffice my lab management? Absolutely no. So as a good laboratory practices, we see that all these resources, etc., are very well managed. And the right side, it is all about various activities that we do, like sampling, weighing, sample preparation, instrument analysis, etc. Various activities which definitely these activities are skilled activities. They cannot be, of course we can definitely understand, but until and unless I do the operation, I will not know how I perform it. So they are like, if I am able to use my both hands in doing them, I would be able to do it properly. So sampling, weighing, measuring, sample preparations, again, a lot of skilled activities they are all. So moving forward, right. So now, as I was telling, there are a lot of instruments, equipments which are used in lab. We have a lot of softwares to operate, those kind of instruments. One such, software is LIMS. LIMS means a lab. You can see here the first line here, a laboratory information management system. So it manages lot of information from the instruments. It is a software and data management basically. So whether it is a balance or whether it is a written procedure, all those in a laboratory are mainly attached to this software, laboratory information management system, and we get the information directly from the LIMS. And definitely, electronic systems will have something called as audit trail. Now this word is very important. Audit and trail, TRAIL, not trial, it is trail, audit trail. So LIMS is a software. Okay. And each of the instrument, definitely, I'm again pretty sure if at all you have handled spectrophotometers in your analytical labs, you must have seen UV probe software, or kind of systems. Definitely there will be a software for us, how to handle, how to run the instrument basically. So similar way, there is chromatography data management system. This is for HPLC, GC, like big instruments, where we have Empower software and Chromeleon software. Now, guys, what do you understand? Why am I talking so much on softwares? Because even if you are preparing your resume, you should know which kind of software you have handled, during your undergraduates or during your graduation, etc. So that's where we should know which software which we have handled. So again, on the computerized systems, like it is just letting us know, this is a completely automated system, whereas here it is a manual injection is done earlier, but now it is automatically the injection. Say I have to give an injection at 6 p.m. in the evening, it will automatically take the injection from the instrument. Whereas earlier, if I have to give an injection at 6 p.m., I have to wait in the lab till 6 and then only give the injection, and monitoring is also to be done. but now it is a software, so it's more compatible, user friendly, all those. Now, computerized systems, very important, part 11. Okay, I was talking about 21 CFR part 210 and 211 earlier. Now I am talking about part 11, which is about electronic documentation. So as we are adopting, digital systems in our industry, we also should ensure that the systems are operating and there is no data misuse, or there is no, if at all backup, the data backups are done pretty well, etc. So that's where here, applicable to the records in electronic form that are created, modified, maintained, archived, retrieved. Retrieval is I have to also not only backup, but also I should retrieve the data also whenever I need them. So we have to ensure that all my systems have this particular ability. So it's, if at all I'm using an instrument, the software which is coming with the instrument should also have these particular features with it. That is what I mean to say. So deletion or unauthorized modification. Now, as we should be very you know clear, or very um, I would say, open. So we guys also heard about data integrity in our classes. If you remember, Shravani was talking about data integrity concepts, etc. So I'll go up less. We will take it again once again. But definitely we cannot delete any data or modify any data. So suppose if I'm testing today, I'm getting a result, and that result is what I'm not expecting. Then there would be, kind of, I may feel, let me delete it, redo it. But in our system, or in any of the pharma companies, we don't delete the data. Suppose if the result is not meeting the required spec, etc., what I will do is I will raise a incident. So there is a different mechanism on handling of these kind of problems, and I will raise the incident, and I will see that what was the root cause, why the result came on a lower side, and then I will, with proper documentation, I go for the retesting of the product. So, I cannot retest or redo the activity without giving in proper justifications. So that is what I am trying to say here. So, and that's where, electronic medium will help us. An analyst who is actually testing the products will never have an access. That is called as privilege. So, will never have a privilege, or in fact, nobody, even the administrator, cannot delete the data. So that's where the privileges are also listed in the electronic computers. So audit trail, review of the trials. So there is a mechanism internally also for us before any other auditor checks our computers. We also have internal audit systems. So we get the audit trail, how the instrument was operating, and then we review it, so that, if at all again, if we find any of the repetition, etc., we have to go why it has happened. A lot of investigation is done during this kind of courses. So electronic signatures, individual logins, and password. Now, if at all you're entering a pharma company, password is again, it's like you should not share. It is just like your bank account details. So your login and password. So those are the few restrictions. 100%. If you're not understanding here, also it is fine. But moving forward, you should understand what exactly is the regulation expectation on computerized systems for us. So deactivation, login IDs, no, never we should do these kind of things. Time stamping, reconstruction of events, no, it's again, a data integrity issue, etc. So, so precautions, log out during shift changes, log in after shift handovers, etc. So our ID and password, that is where we the precautions are talking about, so that I only can use the system. I cannot see, I'm going for lunch. I cannot hand over to, house ma'am to do my injection or do my testing, etc. So that's important, very important in quality control, especially. Again, I repeat, we are in the manufacturing of medicines for patient safety. So it is, as a quality control employee, it is my responsibility that I ensure that the medicines are meeting the specifications, and they are released to a patient, and he is, absolutely healed from the whatever trauma he has. So don't share password kind of things. Now, here again, a brief interest for us on audit trail. So, audit trail is nothing but a chronological, sequence. We can generate this in the instruments to understand, who created the record, who modified the record, or who deleted it, who, when, here are some good words for us, who, what, when, and why they have done that. Suppose I'm injecting, I'm doing a testing on one of the product, diltiazem. Then when I'm, entering into the software, I use my user ID login, and I say, I'm doing assay. So those kind of dialogues will be there in the system, and it can, everybody can understand who were operating the instrument, etc. So that's a very important one on audit trail. So standalone systems, again, all systems, all instruments, all systems is all instruments. Like say vernier calipers. High automation for a vernier calipers is really, does we cannot see, vernier calipers operating by a computer. So those are like, single time, or standalone systems. But even in those kind of systems, vernier calipers, we have a, digital display on the vernier calipers, and we have instrument ID to the vernier calipers. We will get the reading as a display on the vernier calipers. So whenever I am documenting it, I have to mention the instrument ID. I have to mention what was the display, etc. So those are like, standalone systems. They sometimes, the data would be stored only in the system. It cannot be transferred also. So in those kind of cases, we should see how the documentation can be done, meeting the compliance requirements. So that's on computerized systems. So I just brought in, the concept of ALCO here. I am pretty sure everybody knows about this ALCO plus. A for attributable, L for legible, C for contemporaneous, A for accurate, O for original.
Sorry, madam. Your screen is not visible.
What is not visible?
Screen.
Screen, huh? Okay.
Ma'am.
I will just. Is it now okay?
No, ma'am.
Have you checked your mail, Maheshwari, once?
Yeah, okay, ma'am.
Yeah, just see if you can project the slide. So till then, what I will do is I'll just try to give you a glimpse of what we were speaking so far. We were speaking about quality control, few definitions on quality control. We were speaking about
Ma'am, it is
various
visible, ma'am.
functions of quality control, the ICH guidelines, country specific guidelines, etc. We were also speaking
But
about
excuse me, ma'am.
regulatory. Ha, Maheshwari?
Yeah, screen is visible, ma'am. You can continue.
Okay. Yeah. So we were also talking about pharmacopoeias, country specific, pharmacopoeias, etc. We just finished about, computerized systems and the importance of a computerized system in a pharmaceutical company. And we were talking about Alcoa Plus. So this concept was already delivered by my colleague Shravani, but rephrase it like A for attributable, L for legible, C for contemporaneous, O for original, and A for accurate. And always, we have to abide by this Alcoa principle. Complete plus defines as complete, consistent, enduring, and available. So this any result report which is released by my lab by me, then I should ensure that it is meeting all the Alcoa Plus principles. That's where it's important. So I have a short quiz here. Anybody who can put the answer in the chat box. What does LIMS stand for? LIMS. Option A, Laboratory Instrument Management System. Option B, Learning Management System. Option C, Laboratory Information Management System. Any responses, Aishwari?
Students, see, madam.
Answer is C, absolutely. Who was that, who shared the answer?
Shiva Reddy, Tirumala,
Okay. Shiva Reddy,
Shiva
good,
Reddy,
good.
Mrudula, Kavya,
Oh, many of them. Oh, great. Interesting.
and
Thanks, guys.
Kavya.
Thanks for following. Yeah, nice. So we have another question for you guys. I will read out the question. 21 CFR Part 11 is applicable to all electronic records that are created, modified, maintained, archived, retrieved, or transmitted except for sampling and in-process samples. So is it applicable for sampling or in-process also? What do you think?
Madam, screen
Yeah,
is not visible once again.
screen is not visible. No? Actually, I'm pretty much not doing anything. Uh, it is, I really don't know. So is it okay?
Answer is A, madam. No,
Ac-
madam. I'm trying to open my
yeah.
screen.
So the answer is not A. Yeah, that's where the trick is. So 21 CFR Part 11 is actually applicable for all electronic, whether, except is where I'm trying to, make you not think too much. So even for sampling and in-process samples, the 21 CFR Part 11 is highly applicable. So Maheshwari, is it okay?
I'm, yeah, now it is okay, madam. You
If
can continue.
if you can project the PPT, that would be better, I believe. Just
Yes,
try to
madam.
download it. Yeah,
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 you stop it, madam.
yeah. If you have opened,
It
then
has opened.
yeah.
PPT has opened now.
Okay, I am stopping the presentation now. Yes. Yeah, I stopped the share. These kind of things happen, definitely we cannot see that it goes as clear as it is. So ups and downs are common. That's there. Yes. Good. Yeah. Can go to the slide view. Yeah. The next question here, audit trail of. Yeah. This was B. Yeah. Next question. Audit trail of an electronic record should. So B. Timestamp, allow reconstruction of events, track the events modification, deletion, etc. Or option D, all of the above. Yes, you're right, Shivareddy. Any other? Yeah. Yes. Good, Stravya. Right. The answer. Yeah, you can go to the. You can give the answer. Yes, it's D. Next slide. Yeah. Now, this is, core area for you guys, chemicals and reagents. I'm pretty sure all of you have handled a lot of chemicals. You must have, even read about the MSDS, etc. Yeah, thanks, Pavan. Thanks, Tumala also. Yeah, Kavya also. Yeah. So chemicals, why do we use chemicals? Absolutely, to carry out a reaction and to get a result out of the analysis. So substances with known compositions and purity are supposed to be used in the lab. So that is where it is. So the purity of the chemical which I am using, it should be very, which also comes with a COA. COA stands for Certificate of Analysis, and those chemicals only have to be handled in our uh, labs. Again, reagent is also a chemical, but there is a slight difference between a chemical and reagent. A reagent is a mixture of chemicals, whereas chemical is a single entity. Reagent is two or more. You must have used, Nernst reagent, or Grignard's reagents, etc. So that's where it is. Next slide. Yeah, volumetric solutions, and yeah, volumetric solutions are prepared. You weigh the sample, but you actually know how much concentration. The word here for us is concentration. You must have also done a lot of work on molarity, normality, etc. in your undergrads. So those, if at all I know the concentration of my solution, then it comes in volumetric solution. Buffer solutions, again, the definition of buffer is which resists the changes in pH. Okay, and we also use lot of buffer solutions in our testings, and we should know how we are supposed to make a buffer. And solvents. Solvents are also used in our laboratory, like acetonitrile, methanol, etc. And whenever we, of course, purified water is also one of the solvent. Uh, Kavya is telling, ma'am, slides are not visible. Okay. Let's continue. Probably it must be a, slight hitches of the network, but
Friends, please check your network issue. Otherwise, you join in the YouTube. Madam, you are visible now.
probably it must be few hitches in the network. It would be okay still. Yeah. Fine. Yeah, you can continue. Yeah, next slide. Yeah. So good practices. Okay, Pavan, you just bear with my voice for some time. you will be able to see the slide again. Select the grades of chemicals as per the test. Very important. We have lot of grades in the system for the chemicals, and absolutely we have to use the kind of grade which is suitable to the test. Like I'll give you an example here. There is something called IR spectrophotometer, where we do the testing with IR. So the reagent which we use here is KBr. So KBr is a chemical, and especially if I'm using it for IR testing, I use a IR grade of chemical. And if I'm doing a HPLC, I'm supposed to use methanol, which is a HPLC grade. If I'm using the same methanol for a KF auto titrator testing, I use a methanol which is a dried chemical. So that's where when this is an important practice in the lab. So
Madam, just a minute.
yeah, sure. Shall we wait, Maheshwari? So we were talking about reagents, chemicals, instruments which are handled in QC. All these terms, you guys should know these kind of terminologies which are used in industry, and that gives you plus plus when you land here. Yeah.
Madam, it is visible now.
Yes, absolutely. It's visible. Yes. Yeah. So for single peak chromatographic test, now we are just we have just dropped what are the good practices while doing the analysis in the lab. So again, it is not something that I by heart, but I have to follow it during my laboratory activity. So for single peak chromatographic tests, we use cost-effective HPLC grades, etc. So special grades, etc. Those kind of emphasis is given in this slide. So I think you can go to the next slide. Suppose the chemical is having lumps, then absolutely stop. Don't use the chemical. Stop, and then get the right chemical, and then only start your analysis. So that's kind of the practice that is followed. Yeah, laboratory glassware. So I'm pretty sure you guys know about lot of glassware which is used during your analysis, or during uh, testing I will say organic synthesis. Handling glassware is again an important, it's a skill. It cannot be done by everybody. Shall we continue? Okay.
Yes, madam, you can.
Yeah, right. So different kinds of glasswares which are used in quality control are listed in the slide. Uh, slide. If you can present the slide for me, that helps. So right from here, if you see beaker, measuring cylinder, Buckner funnel, burette, the important thing is we should know the word. I have seen cases, especially with the freshers, they cannot say the name of the glass that they are using, like say Erlenmeyer flask or volumetric flask. What is the difference between Erlenmeyer flask or volumetric flask? Those kind of things. We should make sure that we know those differences in the earliest of our career. So
Can you reload? Your voice is up and downing.
okay.
Just Control R.
Is this fine? Hello? So is this fine? Hello?
Yeah, madam, you can
Yeah.
continue. You can on your camera.
So it's the evening call. We have a namaz going on outside. So so a lot of glassware which is listed here, and we have also written the purpose for which it is being used. So you guys should know the terms again. Desiccator, LOD bottle, crucible, TLC chamber, motor and pestle. These right words should be known by people who are working in QC. So next slide. Yeah, laboratory glassware again. Yeah, you can move on. Yeah. So pipettes, we have given few glimpses. We will not play the audio now. We will see if time permits. We can play this. Next slide. So the handling of glassware is important. Always wear what do you say? Yeah. So always wear your gloves and see the labels, marking, etc. is very important whenever we are making solutions. Uh, if we miss this particular upper meniscus, lower meniscus, etc., we will not be able to.
Madam, your voice is not at all visible.
Okay. So anyhow, Maheshwari, I think after this slide, Kaustubh will handle the session.
Okay, let madam to once reload. Kausi madam, please reload your screen. Yeah, you can continue,
Yeah.
ma'am.
Hello, Kausuma.
How to reload? Can you just help
Yeah,
me?
just press Control and R.
Control R.
You just
Okay.
refresh now. Okay. Yeah. So next slide.
Reload.
So this is again on the kind of glassware that we use, 50 ml. What are the lot of information on the glassware. We should know what exactly is that information. So again, I tell you, these kind of informations you can check back, and you can think and Definitely, when you are landing into the lab, we will help you in understanding those kind of terms. Next slide. Yeah. So glassware selection, plus or minus or 0.10 ml, 20 degrees centigrade. Just look over, whenever you are handling, do some exercise. Okay. Next slide. Yeah. Which class of glassware shall be used for volumetric applications in a quality control laboratory? Actually, ideally, we are supposed to use a class A category of glassware. So the answer here to the question is A. So you can take it. You can give the answer. Yeah. While pipetting out using transfer pipettes, the last drop, sticking to the pipette shall be, actually, I will give the answer. It's gently touch the wall of the volumetric flask. So this is, as I told you, it's all about your skill. How do I hold the pipette in my hand? How do I drain the solution? How do I, whether I'm using my thumb, left thumb, or right index finger, or kind of. So these practices should be done very diligently because my results are based on the kind of sample preparation that I'm doing. So absolutely, I halt here. So I hand over the session to Kaustubh ma'am.
Okay, Lakshmi.
Yes, ma'am.
Yeah, I hope I have your support for any questions because from practical point of view, I will explain all the theoretical aspects that are going to for the oncoming slides.
Yes, ma'am.
So, is Maheshwari continuing to present, right? I mean, I don't have this with me.
Yeah. So will you do it, Maheshwari, or shall I do it?
I will do it, ma'am.
Yeah.
Hello, learners. I'll take on from here. My name is Kaustubh Kamath, and I'm from PhD. I'm also working with TTO, Technical Training Organization in Dr. Reddy's Laboratories. You want me to switch on video because that may actually make it difficult for the bandwidth is not correct. But if you want, I can do that. Perhaps on listening mode, it may be more supportive.
Okay, ma'am. You can continue towards the end. You can.
Yeah, I'll just switch on for some time because for the purpose of explanation, it may help because it uses more, I think,
Yes, ma'am.
whatever the technical term is for. So we are now on to pretty much at the end of the session, just maybe 30 minutes to go. So we are on to laboratory standards. So Lakshmi has already explained to you there was a flow. She introduced to all the terms, and then we come to actual analysis, the practices, and all that. And of course, now this is also you can look at it as more as information definitions. Almost all of our analysis is against a certain standard. Like you cannot take something in isolation, one thing, put it in the instrument, and just think that, and whatever result comes out, you cannot just say that yes, this is what it is. It is always comparison. So almost all the analysis is comparison. So that comparison is made against something called standards, and these are the standards. They're called as reference standards. And now here is the definition. You can, I hope you can see the screen. So this can be for the drug substances. It can be for excipients. Excipients are those which go into the formulations other than the other than the active pharmaceutical ingredient. So you need a lot of other things. You may need a stabilizer, you may need a suspending agent, you may need a solubilizer and fillers and etc. So those are all categorized as excipients. Also, of course, the drug itself will come with certain excipients, impurities, correct? You must have seen already from your experience that whenever we manufacture anything, whenever we prepare anything, some small impurities always present. So there are standards for those impurities also. Now the drug over a period of time can degrade. So therefore we need standards for those degradation products also. So I hope you're able to imagine the mammoth scale at which all this is actually handled, which means you need to degrade your drug and get the degradation standards, and throughout analysis you need to use these standards to see whether your drug or drug product etc. is stable as giving what result is expected from that particular product. So and here are the types. One we categorize as primary standards. Now, primary standard is by ICH Q7. It says that it is a substance that has a traceability. So it can be from the pharmacopoeia or another source, usually pharmacopoeia, but probably not exclusive. And it is officially recognized source agency, and it has a traceability, which means all certificate of analysis, where it was produced, what was its characterization, its quantification, its assay, everything is available for that, and that is known as reference standard, and it is categorized as primary standard. And then there is a secondary standard. Now, based on this reference standard, we can actually produce a secondary standard. We have to characterize against this primary standard, and we can use it for our day-to-day analysis. So that would be called as secondary standard. Now, the in-house standard is not defined over here, and it is not there in the next slide. I will briefly explain, and I'll also ask Lakshmi to give in any input if I have left it, which is usually but not exclusively in-house standards are where there is no pharmacopoeia standard available. There we can go for in-house standards. Mostly for impurity standards, there may not be a official standard that is available, so we can actually prepare our own impurity standard, but it has to be characterized and it has to be completely against a certain set of available, whatever is available in the literature or from the from wherever we either follow a pharmacopoeia or we follow some other standard, right? So it has to be characterized against that standard. So Lakshmi, I'll just pause here and ask if anything is to be added for in-house standard definition.
No, ma'am. Absolutely, it is on the characterization. Where in-house standards, we characterize. We go up till NMR, GCMS, etc. Also.
Right. Yeah.
You're right. Yeah.
Then those standards will be taken. Then we are, and these standards, we should not forget that these standards, then we have sample, right? We have sample, we have a standard, and these two together go into for analysis. So how do we now, for analysis, what do we need? So analytical devices, here there is a good classification. One is categorized as instrument and another is categorized as equipment. Instrument is something that will directly deal with the analysis of the substance. It could be excipient, it could be impurity, it could be a drug. But whatever analysis we are performing, it will directly, it is measurement. You see, I'll read out the definition just once. Devices used to collect data, analyze and measure instruments. Instruments used for physical parameters like balance, refractometer, polarimeter, even those. Instruments for chemical quantitative analysis. So if we are measuring, we are characterizing, we are identifying, whatever instrument we are using, all that is categorized as instrument. But for all this, we may need some support also. A lot of support may be required. You may have to filter, you may have to centrifuge, you may have you need air systems, you need some water systems, you need ovens. So all those are categorized as equipments. So that is roughly what this slide talks about. So anything else here? I think that's about it. Yeah. Coming to instrument or equipment, there is a term now. You have an instrument at your hand, right? Let us take a very simple instrument, the simplest of it, titration, which you must be doing. So every day you come in, you use that, you fix the burette, and you start titrating. But when you are dealing with, for example, a pharmaceutical industry, any industry, then is this burette delivering? It is 50 ml burette. Is it delivering 50 ml or not? Now, of course, it doesn't change, right? It is all glass. How does it change? But there are some rubber tubings. There is a wall that delivers certain volume, and all those can sometimes malfunction. There may be some expansion if it is a rubber tubing, and so on and so forth. So therefore, all this must be always you must be sure that whatever instrument you are using is designed to deliver. Instrument is supposed to do its part, and that part, whether it is doing every time, not even once. We cannot say, oh, it did not work for that analysis, that's fine. No, every time, whatever it is supposed to do, it has to do. So we have to make sure the instrument is going to do what it is going to do. It doesn't have a mind of its own. So unless something is going wrong, it may not. It will just continue to function. So our job is to make sure that it is continuing to function, that instrument. So terms we use with that kind of assurances. One is calibration, very important term, calibration. Now, calibration for different instruments we can perform at different times. In some we perform within 3 months. In some it may be daily, like pH meters, for example. And in some it could be 6 months. So I have not come across beyond 6 months, but it could be, but usually 3 months, 6 months, and sometimes monthly. It depends on the kind of instrument, what it is delivering, how long it can continue to function, the way it is supposed to function. It all depends. Those are the criteria that will decide what is the calibration frequency. So calibration is important. Something you can see here appearing in red. So it is simply we are evaluating a set of predetermined parameters against predefined traceable standards and more accurate instrument or acceptance criteria. So there is a set of defined. I will just ask a question before elaborating. Suppose you had to calibrate a thermometer. If somebody can just put in the chat box, how would you do that? You're supposed to calibrate a thermometer that you're supposed to use for that instrument. How do you think you will calibrate this thermometer? Or even simple question, you have to go ahead and use this balance. And I tell you, can you calibrate this balance before use? I want to know that this balance actually weighs exactly as it is supposed to be. So what is the simplest way we will do this? Anybody's answer? So I see a question for a question here. What is it?
I have written the question, ma'am.
Okay.
You can pause for a while.
Okay.
Yeah.
Okay, comparing for standard temperature for thermometers. Yeah, so you want to see whether the thermometer, okay, you are coming a little close. So in case of balance, you will have to use standard weights, right? You will have to use standard weights. So the standard weight is supposed to deliver 50 grams. And then when you place that standard weight, the balance should show 50 grams. Then you are assured that your balance is functioning. Same way, if you want to check on thermometer, if it is showing 100 degrees, whether actually it can detect 100 degrees is what you need to test. So you will be using another thermometer which is calibrated and which is a standard thermometer, and which is like a standard, and that will be also placed along with whatever you want to, if it is a thermometer that is present in your instrument, and then you will see whether your instrument is also giving the same reading. So yes, that is what that's pretty much the principle behind calibration. In calibration, the second was you saw. Can we just go back just 1 second in the previous slide? You see verification. So you can easily see the difference. Just a minute, please. Yeah, sorry. Can we just go back to the slide? I can't see the slide here.
She just minimized it now.
Yeah.
You can continue to explain now.
Yeah, so verification is the difference. Verification also we do pretty much the same thing, except that in calibration, if you find out that your particular whatever you are calibrating the apparatus, if it is not up to the mark, you will actually make the adjustment so that it comes up, and again you will test it again. But in verification, you simply verify and give the result. You do not then make any adjustments whether this is working as it is supposed to work or it is not working. So that is the difference between verification and calibration. I'll wait because perhaps the students would like to see.
Ma'am, we have a question here from Mr.
Yeah.
Kotin.
Is pH meter calibrated with solutions of definite, standard and pH? Yes, it is. So pH meters are calibrated with whatever range we are going to measure the pH. So suppose we want to measure today 5 pH, then we must use at least 4, and then we go up to 6 or 7 standard or 8, so that the 5 should be covered. So use 2 or 3. We also do linearity. So yes, they are done by standard solutions of definite pH. So this was the slide. We just completed it. We can go to the next one. Then we have some other very similar tests, the other term, system suitability tests. And here, Lakshmi, I would like you to add. Now, system suitability tests, we use, at least as far as I am aware of, we use it with chromatography and some higher instruments. And the big difference is that it will be continuously monitored throughout the instrument, throughout the analysis, and also after the analysis, and that is termed as system suitability test. Actually, I'll just pause here.
Yeah.
Anything you need if you want to add?
Absolutely, ma'am. The system suitability test is where we ensure that the instrument was running from the starting till the end without having any problem. And so absolutely.
So we don't do it for an instrument like IR, for example,
Yes.
right? We do it for chromatographic, where there is a long analysis. That's where we need to make sure that all the complete analysis is under control.
We do this for even UV visible spectrophotometer, where we have 12, 24 samples to be analyzed in a continuous stretch. Then we do it.
Okay. There also system suitability tests are performed. So there is another term you can see here, bracketing standards. So these are, of course, which are introduced at the start of the analysis and also in the middle and then at the end. So anybody can write here what is it doing basically? What purpose is it serving? I don't know whether they understood the question though. So when we have, say, for example, 6 samples, right? So we put one bracketing standard, and we know what results are we supposed to get for this, and then we have our sample. Now the sample, whatever the system says, that is what is the result for the sample. So we want to make sure that whatever the system is saying is correct. So but for the bracketing standard, we know what are we supposed to get. So when we inject the first one, we now know that whatever it is saying, whether it is matching the specification, and then we go ahead with the sample, and again in the middle we might give another, and then make sure that whatever next 3 samples have that were done by the system is still continuing to function well, and at the end of the analysis again we finish with another one, so that we make sure that throughout the analysis the instrument has performed as it is expected to perform. Lakshmi, there's a question. Can you take this? Why are bracketing standards used? To unknown concentration, I'm not able to. Oh, okay. You have given that question.
Yeah.
Okay. 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 So Lakshmi has put an answer also to ensure repeatability and reproducibility of the instrument. Okay. What's the answer for this? I'm sure they will get this. Are you all able to see the screen? There is
Yes.
a knowledge quiz here.
Which of the following are considered as primary standard? Option A, USP reference standard. Option B, certified reference standard. Option C, international reference standard. Option D, chemical reference standard. Option E, all of the above. We have one answer
Have you
as
got
D.
D?
Yeah.
D. It's E. Yeah, another answer came as E. Okay. I think there's one more question, but this question I did not see any information in the slide. Let us see whether they can tackle it. Okay, Maheshwari, we can go to the next question. Oh, match the following. Okay.
So calibration, system suitability, preventive maintenance, analytical validation on your left. And we have analytical method performance, documentation, proactive maintenance, performance verification of before start of the analysis, measurement of accuracy before use of the instrument. It's a match the following. You can try to answer one or two.
You can consider A, B, C, D for the answers. The answers that are there in blue or on the left side, sorry, right side. Okay, we got 4A. Good. Then we got 1D. Shravya has given 1D.
Yes. Yeah.
Anything else is coming along?
No. Yeah. Go ahead, Maheshwari. Open the answers.
Yeah, okay.
So preventive maintenance is proactive maintenance. So the next slide. Yeah, it's on analytical techniques, which is keen in analysis. Definitely you guys know about these words. Hold on. Yeah, ma'am. Continue.
Yeah. So we are on to the last part. So we have instruments and we have equipments, but we do need something to go on to perform. So there is some document that will tell us what to do. And so we have three types. One is, we call it analytical methods, right? We need a method, and that will all come under analytical methods. So it will tell us detailed step by step what are we supposed to do for this particular test. And it goes, we have one for the instrument, operation of instrument, which we call it as standard operating procedure, which is not appearing here, but we also have standard testing procedures. Now this will come for each particular product. And there is analytical method, HPLC. What are we supposed to do for this analytical method, and what are the steps you will be following for that. So it deals with methods for determining the chemical composition of the given sample. So there will be lots and lots of them. Each particular substance, whether be it excipient or whether be it a drug, we will have an analytical method available for that. Now, analytical methods for qualitative analysis and quantitative analysis. Now, as the name suggests, if you are going to just say what it is, that is qualitative, which means we're not going to give quantity, exact quantity. We will just say this is paracetamol, this is ibuprofen, this is so and so excipient, or not just even the drug. Maybe just want to identify a particular part of it. That is also part of qualitative analysis. That's very important. Identification tests, limit tests, which there are certain impurities present and we want to do some semi-quantitative. Of course, limit tests are semi-quantitative, but there again we may have some qualitative tests inside. So it can be for impurities, it can be for other substances that are present, the pH of a substance, the given substance, all those come under qualitative. Quantitative, as the name suggests, we are going to give how much of that given substance is present in the sample. That would be quantitative analysis. So I will just read out, provides numerical information of analyte, and analyte is here in the brackets defined as quantitate the the whole thing is in the bracket, quantitate the exact amount or concentration. Whereas for qualitative analysis, provides information about identity of species or functional groups in the sample. In other words, an analyte can be identified. No quantification or amount of the constituents or assay. So everybody knows what assay is. Have you come across the term assay? So assay is amount of substance present in the given sample. So if I give you a sample of aluminium hydroxide and you perform assay as per the given method, the method will be given to you, and at the end of it you will calculate the amount of aluminium hydroxide that is present in the given sample, 99% or 99.5% or 100% or 98%, and that would be considered as assay, the amount of substance, pure substance that is present in the given sample. Here is a detail on qualitative analysis. Identification of the constituents. We can identify functional groups or elements, different elements present. Sodium stearate, the stearic acid component is can be identified, and magnesium can be identified separately. Analysis, measurement of physical attributes or physicochemical parameters of the substance. Before there is a slide on what physical attributes we see, so it will be clear over there. So these are the types of what this slide is telling you about, this different types of qualitative analysis. Here it is, physical methods of analysis. Description is a type, solubility, color and clarity of the solution, melting point or melting radiation, flame coloration, congealing temperature, loss on ignition, residue on ignition, loss on drying, that is here that is a short LOD, PSD, that is particle size distribution by sieving, dimensions, friability, uniformity of weight, hardness, bulk density, tap density. And Lakshmi, have we covered everything, or but this is seems a pretty exhaustive list to me. There may
Actually,
be maybe one or
yeah,
two, but it is quite exhaustive,
we
right?
our the reason we with which we kept the slides here is we wanted them to know what are the various tests terms. Like
Yeah, right.
we will
Terms.
not go into
So
detail.
just we'll pause, and anybody wants any information on that physical attributes, any term that they want any more clarification. Okay. Then we have chemical methods of analysis. Chemical methods is usually over here it is limit test identification, thin layer chromatography. Manual titration also should be chemical method of analysis according to me. So again, you just have introduced certain, just as an example, I believe. So there could be quite a few, but yeah, these are the main ones, right? Limit test. So usually there is a interaction. Whenever there is an interaction between the sample and the reagent, and we are identifying, then that would be considered as chemical method. So, but identification does cover IR. Would we put it under chemical method of analysis? IR? I think it is, yeah, it is coming under instrumental. So I hope that was clear the previous slide. Whenever there is an interaction between the sample and the reagent, we put it under the chemical methods.
Absolutely.
Here are some examples, not all, these are just some examples of instrumental methods of analysis. So pH by pH meter, refractive index again using an instrument, optical activity and and based on that then measurement of specific optical rotation, that is by polarimeter. The instrument that is used to measure is known as polarimeter. Then IR identification by the instrument, which is also known as IR instrument, basically infrared. And then UV visible spectrophotometer. The method is known as UV visible spectroscopy. This is just a few examples. There are many others. Okay, HPLC, GC, and so on and so forth. All those are come under instrumental method of analysis. Okay, Yashoda has put some trail or attempt. Is this a question to answer?
Trying to understand her question. So
Yeah.
probably she's asking about audit trail and trial. The word trial and
Audit trail.
trail. I think.
Yeah. Okay. The difference maybe.
Trial okay. Trial is some.
Audit
Yeah.
trail. Yeah.
So there are two words, Yashoda, audit trail and audit, actually trial. Trials are, of course, attempts kind of thing. But audit trails, trail is a sequence of events which are happening. So that's where there are two different words. Spelling, you should be checking the spelling clearly.
So when we investigate something, then we have to set sequence of events. What we did first, what we did then did second, and at the end then make sense of everything that we have done, and that is known as trail. So we set up complete sequence of events that was done from start to finish. So it is always called as trail, and trial is we already know attempts or court trial, and so that is a different word. So I've just switched on my video for a while because I thought
that helps.
not support.
So
We are at the end of the session. Any general questions? Any more questions?
Students, anyone having any queries, please put in a chat box. Please respond. Today feedback form will be sent by mail. Please check it.
Usually,
Students, anyone
are we
having
supposed to
any?
get more this participation? I don't know. I was under impression that we'll get.
Yeah,
Or,
more participants only is going on, ma'am. In YouTube, just a minute,
ah,
I'll tell.
so
In
some.
YouTube, 700, 800 students are going on, ma'am.
So they have access through YouTube, is it?
Yeah, live section is going on, ma'am. Facebook and YouTube. In Facebook,
Oh,
140 students are there.
okay,
YouTube, 820 students are there.
okay.
820.
So
In
this
air meet,
subtracting us 3, then we have about 15. So what is the difference? I just wanted to understand this. 15 are attending through what, and those 800 students are attending.
not that, ma'am. They are going to live section, madam. Air meet won't come for students.
Okay, I understood. Okay. So they are accessing through a different because
Software.
they don't have access to software, right? Okay.
So we have one question here, ma'am, from Yashoda. She is asking for doing an assay test, how many trials are done usually? So
Okay. Are you asking this question from validation point of view or just for actual analysis?
let's think it as actual analysis and answer
Net
the question.
analysis. Yeah.
So basically, Yashoda, whenever we are doing an assay testing, unlike your lab, you guys do three trials titrations typically, and you get the average out of it and you report it. So similarly, based on the method that we take up, few of the cases we happen to do two sample preparations and then we load it onto the HPLC or UV, etc. But there are cases where only one sample preparation is done and that's it. The result, whatever is from that one one assay, that is considered and we release based on the one particular. So this is all about based on the validation results and based on the data which we already have. So that's where we figure it as one analysis or two analysis, etc. So next question. Difference between polarity and non-polarity compounds. Yeah, ma'am, you would like to take it?
Yeah. So if it is non-polar compound, we use actually there are a lot of details, but in general, if you have a non-polar compound, you will be using a reverse phase um, uh, high performance liquid chromatography. So we have one normal phase and for one reverse phase. For polar compounds, we tend to use normal phase, and for reverse phase for polar compounds, which most of the drugs are, we use reverse phase. And then again the reverse phase and normal phase, then you have difference in the column. So column and the mobile phase that you are using. So again, putting it in very short terms, in reverse phase you have a non-polar column, and for normal phase, for reverse phase you have a non-polar column, and for normal phase you have a polar column. Okay, what's the difference between quality assurance lab of API and formulation? Do you mean quality control? I think. Not much actually. You see, it depends on the tests. But formulations, some of the tests will be common. The lab will look very much similar, but the tests performed, there may be lot and number of tests increase in a formulation. For example, in formulation there may be some other tests that get added up. Whereas for and some tests may not be there which are done for API. For example, maybe the particle size distribution may be done for API and not formulation. It is already formulated. So So some tests are performed exclusively for formulation, some tests are performed exclusive for API, and some tests are performed for both. Assay will be performed for both formulation and API, for example. So impurities or substances and water content, all these may be common. So the tests, that the difference is only what tests they will be performing for formulation and what will be performed for API. Anything else you want to add, Lakshmi can add.
No, ma'am, absolutely fine. We have another question. Which is the right parameter to judge glass apparatus to be used in case of QC purpose?
Okay, I
So,
think you will
yeah, I will answer
this.
Yeah. So basically, the category of glass that we use is class A in laboratory, and absolutely it comes with a certificate of analysis. We take that as the base, and what happens is during our regular usage, we randomly pick up a few of the glass apparatus, say pipette. Suppose we got a lot of 100 pipettes today, we take randomly around couple of 10 pipettes, and we do an in-house calibration of those pipettes, we by using purified water and density method. We check, say the 10 ml pipette is absolutely delivering that 10 ml volume or no. And similar way for the beaker or measuring cylinder. So we have set it up, an internal calibration in, labs where the glass apparatus would be calibrated again, even though it comes with a COA. So that's that question. And we have another question, ma'am. Is it we have to calibrate all the instrument which we have?
Yes,
So,
I don't think there is anything we do not calibrate. Everything that we use, we need to know that it is going to function the what it is supposed to deliver, whatever it is. Even pipettes, even glassware, everything, right? We do. We may not do it throughout for glassware, but at the once when we bring, for example, dissolution apparatus is a good example, right? So whenever we purchase glassware, then we first do check. But for dissolution apparatus, for example, the bowl is actually checked. When we first get that instrument, we make sure that it is 900 ml. If it is 900 ml, it's delivering 900 ml and everything we check. Now, if we have replaced the bowl after, say, 6 months of use for any reason, we will again check whether the bowl is actually delivering or it is holding 900 ml. The mark says 900 ml, whether it is 900 ml. So yes, everything is calibrated,
even
checked. Some will be done only once, and until they are replaced, they may not be done. It doesn't make any sense to keep on checking probably a pipette that has been once checked. It does not change its form or shape. But some others we do periodically. As I said before, some can be done. pH meter is calibrated every day at least once a day at the start of the and before the beginning of the analysis. Some may be for a month, some may hold good for a week, some may good for usually 3 months or 6 months, maximum 6 months. I'm not aware of any calibration that probably dates beyond 6 months. Am I right, Lakshmi?
Absolutely, ma'am.
Yeah.
You're right. So one more question, ma'am. It's a little crazy, but Kavya is asking this.
In QC?
In QC, which department is most important?
Okay. Well, I wouldn't call it any particular department is the most important. Everything is probably in all adding to the overall functioning of the quality control. But still, it's a good, interesting question. So let me try to ultimately look at what becomes eventually the most important function. But from my point of view, everybody has to do everybody's job, but really looking at the results, the analysis of the results, the statistical, ultimately looking at statistical functioning of our, what results we have, which is called a statistical analysis. Would be the most important. It does a lot of things for the QC department. If you understand how many samples came, how many samples got tested, what is our testing time, what is our TAT, what is, how many have we released, what is our failure rate, all these things that is done exclusively using software and analysis. I would call that a very important activity of QC. It doesn't mean that all others are not important, definitely, but that plays a very important role.
So no more questions. I think it's the end. So Maheshwari, can we close the session?
Yes, ma'am. We can close the section.
Yeah. Thank you so much for the
Thank you so much,
opportunity.
madam.
Okay. I hope you enjoyed today's session and wish you all the best.
Yeah.
Thank you so much, madam. Thank you so much, Lakshmi madam, Mani madam. Thank you. A valuable time has spent with us. Really, thank you so much. From me and from our organization and from students also.
Yeah. Thank you, guys.
Thank you so
Yeah.
much.
Have a nice weekend. Bye.
Bye bye,
Okay,
ma'am.
bye.